Today we'll be learning about blood as evidence. We'll learn about the components of blood as well as how blood typing is used in forensics and some of the tests that we use to determine if the substance we found even is blood. So let's get to it. Blood evidence can be found in several different things: blood samples, blood droplets, and blood spatter. Blood samples can be analyzed to determine blood type and DNA, which we can match to suspects or use to eliminate suspects. Blood droplets can give us clues as to the location of the crime, the movement of the victim, what kind of weapon was used. Blood spatter can be analyzed to determine patterns to help us figure out how the crime happened. Tests for blood are so sensitive that they will expose the presence of blood even after a surface has been wiped clean, and we'll talk about some of those. Blood that's many years old can still be identified as blood and analyzed for DNA. Let's first talk about the components of blood. Blood's a mixture of water, cells, enzymes, proteins, and inorganic material. Plasma is the liquid portion of blood. Cells, among other things, make up the solid portion. And the three types of cells are erythrocytes, which are red blood cells, leukocytes, which are white blood cells, and thrombocytes, which are platelets. This is what happens when they take blood and they centrifuge it. They spin it. It divides into the different parts. So the erythrocytes make up about 45% of the blood. Platelets and leukocytes make up about 1%, and plasma makes up the rest. Red blood cells have identifiers on the surface of them. There's millions of them, and they are called antigens. An antigen is a protein that can stimulate the body to produce antibodies against it or stimulate the immune response. And these antigens are responsible for these blood type characteristics. Is it A blood or B blood or AB? And they're grouped into systems. This picture shows some red blood cells with their antigens. An O negative person has no antigens on it. An A negative person has A antigens. A B negative person has B antigens. AB has A and B antigens. So we have A antigens and B antigens, and the combination or lack of those is what tells us what blood type they are. We also have Rh antigens. That tells us whether we have positive blood or negative blood. If you have the Rh antigen or identifier, you have positive blood. If you don't have it, you have negative blood. Antigens allow your body to create a defense against future invaders. Antibodies circulate in your body once they are created to identify, attack, and destroy the same type of antigens if they enter the body again. The antibody will only react with its specific antigen. So there's an antibody called anti-A antibody that would identify, attack, and destroy A antigens. We have an anti-B antibody that will identify and destroy B antigens. Antibodies are what we call bivalent. They have two reactive sites. This means that each antibody can be attached to antigens on two different red blood cells. This creates a network of cross-linked cells called clumping or agglutination or clotting. It wouldn't be good for blood to clump together in veins, so type A blood has no anti-A antigen. Type A blood does have anti-B antigens. So if a person with type A blood got a type B transfusion, they could suffer a reaction and die. Here are the characteristics of these different blood types. If you have type A blood, you'll have A antigens or identifiers, and you'll have anti-B antibodies in your blood plasma. If you have type B blood, you'll have B antigens and anti-A antibodies. If you have type AB blood, you'll have A and B antigens and no antibodies. If you have type O blood, you won't have any antigens or identifiers, and you'll have both anti-A and anti-B antibodies. Why do we care about blood typing? Blood typing is often used to eliminate suspects. It's cheaper and it's less time-consuming than DNA profiling, so it may be something that's used first before we go to DNA. Here's a different visual of different blood types and their antibodies and the antigens. Doing this blood typing has limitations, though, mostly because of its lack of discriminatory power. What that means is that a lot of people can have the same blood type, making it only class evidence. So blood typing can put someone in a category of people who have A blood or people who have B blood, but it can't be used to identify an individual. There's also a potential for inaccuracy. We may have false positives or negatives due to degradation of the blood. Age, weather, and putrefaction, which we'll learn about in another unit, can degrade the blood samples, making typing unreliable or inconclusive. Samples can also be contaminated by other blood, other bodily fluids, or microorganisms like bacteria from the skin, leading to false results. When we found something that we suspect to be blood at a scene, we have to answer 3 questions. First, the investigator has to determine whether the suspected blood is blood at all. And they do that using several different blood reagent tests or presumptive tests, which we'll talk about next. The purpose of a presumptive test for blood is to determine whether or not it is likely that this body fluid is present. In other words, the positive result indicates the possibility of blood. Once they've determined that it is blood, then they need to determine whether the blood is human blood rather than the blood of an animal. After determining that it is human blood, investigators will then try and associate the blood with an individual, first by performing blood typing tests to rule out suspects, then by doing DNA analysis on the blood if possible. Now we'll talk about some of the tests that we can do to detect blood at a scene. The first is the light source. Investigators will first examine the crime scene to look for areas that may contain blood. They may use high-intensity light or UV lights to help them find traces of blood as well as other bodily fluids that are not visible under normal lighting conditions. They'll also use blood reagent tests, referred to as presumptive tests, to detect blood at crime scenes based on the properties of hemoglobin in the blood, and then further tests at the crime lab can determine if it's human blood or not. Phenolphthalein is a chemical that is still utilized today and is usually referred to as the Kastle-Meyer test and produces a pink color when it reacts with hemoglobin, and I'll show you this in class. Hemastix is a strip that has been coated with tetramethylbenzidine or TMB, and it will produce a green or a blue-green color with the presence of hemoglobin. Hexagon OBTI is a test that displays positive reactions for human, primate, ferret, and skunk blood. Why is that useful? It at least narrows it down for us. Luminol or Blue Star are chemicals used by crime scene investigators to locate traces of blood, even if it's been cleaned or removed. Investigators will spray a luminol solution throughout the area under investigation and look for reactions with iron present in the blood, which causes a blue luminescence or glowing. One problem is that other substances also react, such as some metals, paints, cleaning products, and plant materials. Another problem is that the chemical reaction can destroy other evidence in the crime scene. This can be used to look for blood in scenes that may have been cleaned or for trace amounts not visible with the naked eye. We can also use it to look on items used to do the cleaning, like sponges, cloths, mops, etc. Fluorescein is a chemical also capable of detecting latent or old blood, similar to luminol or Blue Star. It's ideal for fine stains or smears found throughout a crime scene. After the solution has been sprayed onto the substance or area suspected to contain blood, a UV light and goggles are used to detect any illuminated areas which appear greenish white if blood is present. It may also react to many of the same things as luminol, like copper or bleach. Leucocrystal violet, or LCV, is one type of chemical process that is used for blood enhancement. Using this test helps to make the blood evidence more visible so it can be photographed and analyzed. Here's some pictures of Blue Star being used in an actual scene. So this person washes their arms off and then cleans out the sink, but using that Blue Star, we can still see traces of the blood. Here's some more examples. And we'll look at this in the lab because it's pretty cool to look at.